All right, so today we're going to start with lecture again and then move into lab. In lab today, we're going to do something very similar to what we did yesterday, but we're going to be using micropipettes, which measure a much smaller volume. And we will also have a problem session. So I have a couple of questions for you guys to work on in groups, which I won't be grading, but it's good to think through these things. And again, it's good practice to find your blind spots. And today in lecture, we're going to move on from proteins and start talking about DNA, which we've mentioned a couple of times already, but we're really going to talk about the entire structure of a DNA molecule, as well as the parts that make up a DNA molecule, which are the nucleotides. And so I always like to begin by mentioning the people who were responsible for discovering the double helical nature of DNA. And Watson and Crick get a lot of credit for this, but I think in the last 5 years or so, some of the other researchers who actually did the research in lab have also been getting more credit. In particular, these two people, Griffith on the left, and then Rosalind Franklin on the right. And again, they were the ones who actually did the research in the lab, whereas Watson and Crick actually did not do any research at all, which was kind of surprising. But they did look at the data and come up with the structure of DNA. And there is a movie about this. It's from the 80s, maybe the 90s, but I think it's really interesting. I had a professor show it to us in virology, I think, and you can find it on YouTube. It's a little bit poor quality, but like I said, it's very interesting if you're interested in the history of science. And so I linked it there. So we've been talking since class began about this schema of DNA as information that is used to make messenger RNA, which also carries information, which is read to make strings of amino acids, which we talked about yesterday, which can be folded into proteins. And this is where all of the activity from a cell comes from, or most of the activity of a cell comes from these proteins, which can carry out different functions. And of course, some proteins are also only structural, but that doesn't make them any less important. And so something I wanted to point out that I'm not sure we really said explicitly is that these proteins are made up of amino acids, which are called or referred to as monomers. So monomers are any type of molecule that is repeated over and over again in a larger molecule. And so as I said, in proteins, our monomers are amino acid. And then the string of amino acids is called a polymer or a protein. And then this is what it's like. But on the right side there, it says a chain of monomers is called a polymer. And so we've probably heard of this word before. And I think that we oftentimes hear of polymers in the context of plastics or other manufactured synthesized materials. But again, polymer is a term that could be applied to any string of monomers put together. So monomers here, and then we have a new box of Sharpies here. And the monomers, again, we have amino acids. And then on the polymer, we have the peptide chain. And then for DNA, the polymer is the DNA molecule. And then what is our monomer for the DNA polymer? Nucleotides, right. And nucleotides are also the monomer for RNA. And we'll talk about the difference between the two throughout the course of this class. And I think that we briefly looked at that slide a couple days ago that talked about the most common small molecules in the cell. So we can also have monomers and polymers of other molecules. So for example, if we have glucose, which is a carbohydrate, and glucose as our monomer, then a polymer of glucose, you can put it together in a bunch of different ways, really. We would have things like starch, glycogen, and even cellulose. And so again, this is just an example. We're not going to talk about the polymers of glucose very much. We'll talk about glucose at the end of the semester. But again, glucose would be a monomer, and then when you take a bunch of glucose molecules and bind them together in a string, you're going to get these polymers. Okay, so moving on, focus on DNA and RNA. So what is DNA specifically? DNA is a polymer of deoxyribonucleic acids. Okay, so that sounds really complicated, but we're just specifying the type of nucleotide that we have in a string of nucleotides that makes up DNA. And again, if we look at the word itself and break down the roots of the word, first we have deoxy, so that means that it's missing an oxygen. And we'll see where it's missing an oxygen in comparison to RNA in what moment. And then ribonucleic acid. And so if we break that down, then it's a little bit more complex if you don't know a lot of chemistry. But we'll see that we have some acids, and we're going to have an nitrogenous base, and so on. And so RNA, the polymer of ribonucleic acids, doesn't have that deoxy part. And here is an image of a string on the left of DNA, and then a single nucleotide is shown on the right. And we see that we could categorize different parts of the nucleotide. And in particular, I want to point out the phosphate group, which is highlighted here, the sugar group, which is in the middle, and then the nitrogenous groups. And this is the monomer. You see these three groups, and we'll talk about the importance of each of them in a moment. But we see these three groups in every single nucleotide in every single monomer of DNA, As we see on the left side there, then we just have these repeating units of this over and over again to make up our entire genome. So we have millions, tens of millions, hundreds of millions of these nucleotides contained within our genome that code for our proteins. So let's talk about these three different groups. So the nitrogenous bases are the parts that are actually what we think of when we say DNA or RNA. These are the letters that we give to identify the different nucleotides. So up in the top row, we see that there's cytosine, thymine, and uracil. So these are different types of bases. And then at the bottom here, we have adenine and guanine. And you'll see that if you look closely at the chemistry, that each one of these has a different chemical structure, and that there are actually two different groups. And you see that the row on top has a single ring structure, whereas the ring on the bottom has a double ring structure. And these molecules are larger. And this is really important. Can anyone think of why? So if you look at, when you think about the abbreviations of these names, you think about the base pairing rules. Why is it important that we have these two different sizes of these nitrogenous bases? Why don't you guys talk to the person next to you for 10, 15 seconds? So the question is, we have two different sizes of nitrogenous bases. Why is that significant if you think about the base pairing rules? So first you might want to write down what the base pairs, the pairs are.
That's H2, 2.2C, yeah. So you can have
Anybody have any ideas?
Is that what you're saying, I guess? Is it maybe because if you pair, like, for example, A goes with T and you pair A with G, then maybe you won't make the proper function?
Well, that's part of it. So if we pair up the wrong two base pairs, then you're not going to have, then you can change the function because the structure is going to be wrong. Comment in back? No? Okay. Right. Right. So, excuse me. As I said, the top row, the pyrimidines have a single ring structure, whereas the purines down below have a double ring structure. So, we take a look at our base pairing rules. We have A and T, and G and C, and then in RNA we have A and U. And for A, we have a double ring structure. For T, we have a single ring structure. And then for G, we have a double ring structure. And for C, we have a single ring. So in the base pairing, we always have a double ring with a single ring. And that's really important because we know that our DNA is double-stranded, looks something like this, when you lay it out flat. And by always having the double ring with the single ring, comparing with the pyrimidine, the width of the DNA is consistent. So if we had a single ring and a single ring nucleotide, then this would be more narrow. And if we had a double ring with a double ring, then it would be wider. And this is what happens if we get some kind of mismatching, mutation, or the wrong DNA or nucleotide put in during DNA synthesis. Then this does alter the width of the DNA helix in that one specific spot. Otherwise, the width of the DNA helix should be consistent. And I always like to point out, so we have these two different groups, and I always tell students, if you remember, pyrimidines have the longer name, but they're the smaller ones, say the single ring, and the purines have the shorter name, but they're the larger ones, and that seems to work well. Okay, so that's the nitrogenous base part of the nucleotide. So again, we look at the nucleotide here, the nitrogenous base, that's what we think of as the essence of DNA, and that's what we read when we're sequencing DNA. The sugar part is in the middle there, that's the entire molecule. And the sugars determine whether this is RNA or DNA. So this is a ring structure made up of carbons. Again, if you had to guess, carbon is one of the most common atoms or elements in organisms. So you would guess that these would be carbons, even though they're not labeled. Just out of curiosity, has anyone here taken organic chemistry? Yeah. So you're probably not used to this kind of notation of drawing carbon ring structures, but whenever you see a ring structure like this that doesn't have any element indicated at the points, that means that there's a carbon there. So in these rings, at each of these points, we have a carbon, except for at the top where we have the oxygen labeled there. And I have another image later on that shows the carbons. So, as I mentioned, we would look to the name of these molecules, deoxyribose versus ribose, to determine the difference between DNA and RNA. And DNA is deoxyribose because if you look really closely here, it has a hydrogen on this second carbon, whereas RNA has hydroxyl, so it has an additional oxygen. And so that's the only difference in terms of the sugar between DNA and RNA. Pretty small difference, but it's very significant chemically. So it makes RNA susceptible, for example, to changes in pH, whereas DNA is much more stable. And as we know, they're used for very different functions in the cell. And so they'll also bind to different enzymes. And so I also want to explain quickly, but I don't want you to get hung up on this, about the numbering on the carbons. So again, if you go on in your lives to take organic chemistry, you'll learn all about the numbering of carbons and rings and things. But for now, you can just memorize the numbering for the sugars So the first carbon is here. If you're looking, you start at the oxygen and then go clockwise. So this is C1, this is C2, and we just go in order. And we have 5 carbons. So C1, we're not so interested in. C2 is the one where we can either have a hydrogen or we can have a hydroxyl, depending on if we have RNA or if we have DNA. So this would be RNA, and then this would be DNA. And we refer to these different carbons as 2', 3', 4', 5', etc. And we're going to come back to this in a couple of minutes when we're talking about determining the 2 different ends of a string of DNA or RNA monomers. Well, as I said, we'll come back to that in a moment. Any questions so far? Is this stuff familiar to some of you? Yeah. Yes, no, maybe. So the final group, I'll show you the whole molecule. Yeah, the final group here is the phosphate group. And we've talked about phosphate groups before. They carry a negative charge and lots of oxygens. And this phosphate group with the oxygens that have these negative charges actually gives the whole DNA molecule a negative charge. So who here has heard of electrophoresis? I know everyone here has seen it in a crime drama, absolutely. This is when you have a gel and you run the DNA on it, and it's sometimes black and white, sometimes the line from the top, and then you have DNA actually moving through the gel. Does this ring a bell? Maybe a little bit. I'm sure you guys have seen this at some point, but maybe not explained actually scientifically. But this is where we use electricity to actually move DNA molecules. Because of these phosphate groups with a negative charge, we can use electricity to move DNA because DNA is going to be attracted to a positive charge. So at the bottom of the setup, we have a positive anode and the DNA moves towards that positive charge. Pretty neat. And also, so keep in mind that, yeah, we make use of this in lab, but also in your cells, DNA has a negative charge, and we're going to see that because of this, it interacts with positively charged proteins. So what would give a protein a positive charge? In the bigger picture. So what are proteins made up of? Amino acids. So an amino acid with a positive charge. We make really easy connection. So an amino acid with a positive charge, there are a couple of them. So proteins with a lot of these positively charged amino acids often interact with DNA, and we're going to talk about those at the end of this class as well. So when we have a string of nucleotides of DNA, then we form this double helix which runs anti-parallel. So what does that mean? Well, double helix is pretty straightforward. It just means that we have 2 polymers of DNA, and the base pair, or the bases, are pairing through hydrogen bonding. And we mentioned this at the beginning of class. We said that the base pairs interact, they have hydrogen bonds, and if you can see in this image, you see that there are multiple hydrogen bonds between each of the base pairs, so that then when you add all of these hydrogen bonds up, you have many more times hydrogen bonds when you have nucleotides, and if you have millions or hundreds of millions of nucleotides, then it's a lot of hydrogen bonds. And because of the structure of the nucleotides, then we have the curling of the helix. Now, what's the other part that I didn't talk about? I said that this is an anti-parallel double helix. What does that mean? So it means that the two strands, the two polymers, are running in opposite directions. So I drew this nucleotide, or this sugar here. On the left, we have something that looks like this in that double helix. On the right side, we take this and turn it upside down. And so this is one of the most difficult things to figure out about the structure of DNA when they were trying to figure it out, when Watson and Crick were trying to figure it out. Because no one really expected this anti-parallel structure. But this is the only way that the base pairs can line up in the middle of the DNA helix and be read and replicated. And so this means that that when we are reading DNA, or when a molecule or an enzyme is reading a strand of DNA, it can go in one direction, but if it wants to read the other strand of DNA, it needs to go in the opposite direction. And this causes a lot of complications during the DNA replication, which we'll talk about next week. It also means that if we're looking at a DNA molecule and we want to give the sequence to someone, we need to know which end to start at. And by which end, I mean the 5' end or the 3' end. And so this is all determined by, or labeled by, the different carbons that we see in the sugar structure. So don't forget, the sugar is not the only thing that we have in a nucleotide, but that's how we name the ends of the nucleotide. Right, so anti-parallel means that they're parallel, but they're going in different directions. So on one side we have this right side up, on the other side we have it upside down. And you see this in this image, which is a little bit simplified, but I think that it's a really good image for really pointing out the anti-parallel parts of the DNA double helix. So here we have the 5' carbon, which is the one that's not part of the ring. And we have the 3' carbon, which is part of the ring. And the 3' carbon of this nucleotide is going to be bound to the next nucleotide. Then when we look at the other side, the right side here, the 5' carbon we see, the one that's not part of the ring, is on the bottom of the strand. If we look across here on the left, the 5' carbon is on the top. At the bottom of the strand, we have the 3' carbon. And again, as I said, I have the numbering here of the carbons. So a little bit more about this, showing a little bit more of the chemical structure. So how can we tell if one end of the DNA is 5' versus 3'? Well, the things to look for, if you don't have the numbers of the carbons, are a phosphate group versus a hydroxyl group. So off of the 5' carbon, there is a phosphate group. So put that here. And then off of the 3' carbon, there is a hydroxyl group on both DNA and on RNA. So So oftentimes people get tripped up because we said on this carbon right next to this, there's only a hydroxyl on RNA molecules. On DNA, we just have the hydrogen. But on this other carbon right next to it, on the 3' carbon, you always have a hydroxyl, RNA or DNA. And this is crucial, this hydroxyl. If you don't have this hydroxyl, then you can't add anything onto this. So if for some reason you had a nucleotide that's missing this hydroxyl, then that would end the DNA strand. There was some kind of
I'm not sure if you said this already, but is there a difference of HO and OH?
No. So these are the same.
So it means the same thing, it's just hydroxyl.
Yes, exactly.
It doesn't matter which way you write it.
No, you could write it either
OH
way.
or HO.
Yeah. So it's usually just convenience for indicating what that molecule is going to interact with. Okay. And so, as I said, this is called a free hydroxyl when it's not attached to anything, and this is crucial for adding on the next nucleotide in the chain. And by knowing the 5' end versus the 3' end and what direction this chain is going, then we know what direction DNA could be synthesized in. So as I just said, the 3' hydroxyl is where we add the next nucleotide. So we say that DNA is synthesized 5' to 3'. Because as we add in the next nucleotide, we're going to add 5' phosphate. It's going to bind to that 3', and then we have a new 3' hydroxyl to this. And as I mentioned, we read in the opposite direction of synthesis. We read out of the 5' enzyme is just reading the DNA. And again, we'll see more about this later as we go through problems. This will become more clear about those things. And so if we were going to say, we were going to determine whether this is a chain of RNA versus DNA. And we changed the structure of this last night. So how do we know that this is a chain of DNA and not RNA? Yeah. Right, so this carbon right here, the 2' carbon, we don't see anything indicated, which means there's hydrogen there. We don't have another oxygen. What's another way that we know that this is DNA? Right, so in DNA, we have thymine. In RNA, we have uracil. So that's a good indication as well. The one thing that could trip us up is that this is a single strand, and normally we see DNA in double strand. But this is just an example, so we could kind of gloss over that. So this is a comparison of DNA double helix laid out flat on the left versus what you would actually imagine it to be in 3D on the right. And again, we see that the strands are anti-parallel and that we have hydrogen bonds in between the base pairs. And this image, both of these images, do an excellent job of showing the hydrogen bonds. There are multiple hydrogen bonds between each of the base pairs. And something I'd like to point out here is that AT pairs have fewer hydrogen bonds than GC pairs.
Is it just this one or is it others?
So whatever you have in adenine and thymine, you're going to have two hydrogen bonds, which would be nitrogen, and then hydrogen, and then hydrogen. And with the structure here, you're always going to have this exact chemical interaction between the G and the C. And so I think that you bring up a good point. Whenever we are talking about going through a DNA molecule and reading the sequence, we're always going to have these exact same chemical structures and interactions. So what we see here, you could diagram out an entire genome just from this here on the left. Because we have the base pairs, and there are only two types of pairs. So on the left-hand strand, maybe we would flip this over, maybe we have thymine on the left-hand side of cytosine, but this is all we need to really understand the entire genome. And I think that this will come up in one of the problem sets next week. But again, to emphasize, this is significant, the difference between 3 hydrogen bonds and 2 hydrogen bonds. So in real life, if you are doing research on an organism that, for example, comes from a very hot environment, so if you have a thermophile and you're trying to sequence the genome, you actually have to heat the DNA to a higher temperature because those organisms have more GC pairs in their genome, and that stabilizes the DNA more. It makes the double helix stronger because you have more hydrogen bonds. And normal organisms would tend to have more AT pairs if they don't require the stabilization. And we talked about this previously, but I can listen to it again. How are the weak hydrogen bonds able to hold together our DNA strands? This is, very important that our strands of DNA stay together. Why do we kind of trust this job to the weak hydrogen bonds? Because there's so many. And again, in general, why do we have, what determines hydrogen bond? So obviously we need a hydrogen in order to have a hydrogen bond. And what else do we need to have? Right, an electronegative atom, something with either a partial negative charge or a full negative charge. And then here, this is just a diagram of what we have on the board minutes ago, which is the uniform diameter of the DNA double helix. And this shows, again, how you have a different width of the DNA if we have 2 pyrimidines or 2 purines, as opposed to 1 of each. Any questions about DNA double helix? So we didn't talk about RNA very much. We'll talk a lot about RNA in a week or two, but I did want to mention that most of the time when we think of RNA, especially colloquially, RNA is messenger RNA, so it's single-stranded. But in reality, in the cell, there's a lot of RNA that is double-stranded. in part because longer strands of RNA can base pair with itself. So that's what we see on the right-hand side. That's a transfer or tRNA molecule. And it's all one continuous strand, but it can fold up so that those base pairs can hydrogen bond with one another. So really similar to DNA, and you see again kind of helical or turned nature to this molecule, because again, the nucleotides are very similar. But this is one continuous strand instead of having two separate strands.
So it's
And to wrap up this section, just as a reminder, what's going on here, what is the overarching thing that we're talking about? We're talking about DNA carrying information that is used to make messenger RNA, and that is read to proteins. So one thing actually that I should mention are, yeah, there we go. So when we're talking about this string of nucleotides, what types of bonds are there between the nucleotides in a single strand? We know that hydrogen bonds hold the two strands together, but there's not hydrogen bonds that are within a single strand. We know that these are all covalent bonds. So the bonds that hold together the single strand of nucleotides of DNA and RNA are called phosphodiester bonds. And again, think of phospho, okay, because we know that the phosphate group is involved, and then the diester part is more if you're confident in your chemistry. But in addition to that being the name of the bonds between the nucleotides, we also refer to the part, the sugar and phosphate group, as the phosphodiester backbone. So if we're referring to the phosphodiester backbone, it means the part of a DNA molecule that doesn't have these nitrogenous bases. So basically just what's shown in yellow on the right, the phosphodiester backbone. So the phosphodiester backbone is the bonds between the phosphate and the sugars.
so the nucleotides.
So the two strands are held together by hydrogen bonds. Right, so each of the nucleotides is bound to the next one through these covalent bonds, which are called phosphodiester bonds.
Oh, so
Yes. So actually, on that on here. Yes, so within each strand. And so if we add this onto our chart here, the bonds between the monomers, bonds between amino acids are called what? Peptide bonds. Peptide bonds. And then the bonds between nucleotides are phosphodiester. And we're not going to get into the bonds between the glucose molecules because that varies a lot depending on the molecule. Yep. So the backbone is just called the phosphodiester backbone. And that involves the sugar group, which is the ring that we drew up here, and the phosphate group, which is up here. So basically I've drawn one piece of the phosphodiester backbone here. The other part that's missing is the nitrogenous base, which is A, T, G, or C, which comes up here. And so that's the only thing that's not part of the phosphodiester backbone.
And those are two.
Exactly. So yes, peptide bonds, phosphodiester, both covalent bonds. And I like this comparison because it really specifies what bonds we're looking at in DNA. Because we know between amino acids, there are those peptide bonds in a single string of amino acids. In a single string of nucleotides, we have phosphodiester bonds. When we're talking about two strands, then we're talking about hydrogen bonds. Yeah.
So, for example, peptide bonds hold together amino acids, right? And then, so that means that phosphodiester bonds hold together or join nucleotides,
Right.
which the nucleotides are the ATPC.
So that is the nitrogenous base part of the nucleotide.
And that's what phosphodiester bonds do. They join together. They join the nucleotides, or what exactly do they?
So up on the right-hand side there, we have the nucleotide. And this part is the A, P, G, or C. This shows a single ring structure, but we know it could be two also, depending on the identity of the nitrogenous base. And then the sugar phosphate backbone involves this part, and we're going to have a phosphodiester bond between this phosphate and hydroxyl on the next nucleotide. So we see that here. This highlighted in red is from the bond, between these two nucleotides, the one up here and the one down here. So we see the covalent bond between the hydroxyl group here off of the 3' carbon and a phosphate group here off of the 5' carbon. The covalent bonds here that it's labeling of the phosphodiester bonds. Another question. So, good question. We could see it here. So I only put 3 oxygens on that phosphate group because the 4th oxygen is going to come from the next nucleotide. So remember again, we're talking about covalent bonds, which means the sharing of electrons. And the sharing of electrons is going to come from the oxygen on this hydroxyl group. So this guy is going to share electrons with a phosphate group from the next nucleotide. So we have a covalent bond that's forming. Well, there's oxygen to oxygen. Sorry. Sorry. Those are making me draw. Camera breaks. So this is what we'd have. So we've talked about inorganic phosphate groups before, where we have a phosphate with, or a phosphorus with 4 oxygens. So this phosphorus is going to end up with 4 oxygens bound to it, but 1 is from the previous nucleotide, and 1 is going to, the other 3, sorry, are going to be from the nucleotide that this phosphate group is a member of. Did that clarify, or did that make it less clear? So again, I can point it out here. So in this image, for this bond right here, this phosphate group is bound to this oxygen, this one, and this one. Those belong to the nucleotide below. This oxygen belongs to the nucleotide above. This is a hydroxy group. It's coming off of 2' carbon. So this oxygen belongs to this nucleotide, 2' carbon. It's sharing the electrons with this phosphate, which is coming off of 5' carbon of the nucleotide below. Yeah, I mean, to be completely accurate, we would draw this, but oftentimes we don't. So I think that you're referring to this image. So we wouldn't even draw this. Oftentimes we would just draw the nitrogen space. Any questions? Let's take a 3-minute break, because I feel like everybody's like, okay, brains are full. So take a breather for a couple minutes, and then we'll start in on some other stuff.
If I can find out if they'll come next time, because I don't know.
Sorry about that.
Oh, you
Yeah.
I was like waiting for you forever, and I
I'm
was
sorry about that. Yeah, I need to update the schedule. I need to be Mondays and Thursdays.
like,
Yeah,
okay,
I'm sorry about that.
no problem. I was just like waiting.
Yeah.
So I really didn't wait, and I was like, I think it's time to go.
And so where can we find the updated schedule?
So I'm gonna post them. I have probably today. All right.
And if you send one to me, then I'll put it on our platform.
Okay.
Did you
Ask
have?
a question? I'm not sure.
I'm not sure if this one gets tested, but I know it's gonna bring up to yesterday.
Okay.
So it's really bad. I'm struggling with it.
Okay. Oh.
I'm very difficult, so I'm not a lot,
Mhm.
but the testing is very, very good. So, yeah. Can you just elaborate on in terms of the structural limitations? Like, I think it's kind of aspirates to like kind of just some of the fine ideas just make sense.
Yeah, so I don't think there'll be anything specifically on the exhibit for that. And I think that throughout the entire class, the only way we're going to refer to structural motifs is saying this protein has a helix turn helix motif, which helps it to bind to DNA. I think that's really the extent that we'll be talking to them, talking about them, not talking to proteins.
It's like a very over the surface.
Right. And the only time that I've really seen those used is, so if you're in a lab and you say, okay, I want this DNA sequence and I want to know what kind of protein it is, then you could analyze that sequence and say, oh, so because of the hydrophilic, hydrophobic, or polar properties of these amino acids, we think it's going to fold this way to make something that looks like a helix to a helix.
So we talked about heat shock proteins, how they bind in says the single plates of folding it. And one of them is chocolate. And then we also talked about denaturation.
Yes.
Can you explain the difference of those two? Is it something
Okay.
they kind of make them fold?
So yeah, denaturing means that the protein unfolds. It could be because of heat, it could be because of high salts, it could be because of pH. All those would just be referred to in general as denaturing a protein. And then some proteins can spontaneously refold. Not many. We saw that one of the RNases can start to refold because of the disulfide bonds. Most proteins need resistance to refolding, and things like heat shock proteins and chaperone proteins would assist in that.
So you kind of take on
They are helixes. And we'll see with chaperones, again, we're talking about making proteins initially, so they also can help proteins fold correctly the first time.
just really want to understand this. For women like it's up to 18 so far. So I feel like we're really getting this down now.
Definitely. And actually, we focus a lot on molecular independence of genetics.
Just like they don't like the things. Detections.
Don't teach genetics. Yeah.
And I guess once we get through this, we'll get on to like actually cell structure.
Yeah. So we'll do that, and then we'll talk a little bit about metabolisms, cellular respiration.
Yeah. Photosynthesis.
Yes. Yeah, and so actually, then we'll come back and end with Mendel and genetics. Because that part is not as molecular, and I think that a lot of people are familiar with it, so I wanted to emphasize that. Yeah. That's very something that'll be. Oh no, there'll be lots of them. So if you already downloaded the slides or if you're following along, I'm going through these slightly out of order because I feel like we need story time now instead of more molecular stuff and chemistry. So we're going to go through some experiments and talk a little bit about the history of determining that DNA is actually the molecule that determines our genetics and carries any heritable traits. And that being said, we're going to talk about Mendel and pea plants at the very end of class because I find that that's something people are more familiar with. So I wanted to end with something less stressful than something more stressful. I used to end with cellular respiration and photosynthesis, and everybody hates that for some reason. So, let's end with something a little bit more comfortable. So we're not going to talk about Mendel right now, but I'm sure we've all heard about Mendel at some point in our lives. He was a scientist, he was a monk, and he did all these experiments with pea plants and said, oh, okay, so traits can be transferred from parent to offspring. And that was in the 1800s, the late 1800s. But decades and decades later, scientists still didn't know what molecule was allowing for traits to be transferred from parents to offspring. And there were two main candidates, which were DNA and proteins. So scientists in the 1940s were aware of both DNA and proteins. They didn't know as much about the structure of either of them as we do now, obviously. And there were some scientists that thought that DNA was the heritable molecule. And by heritable molecule, we mean the molecule that is transferred, again, from parent to offspring, and carries some kind of genetic information. So heritable molecules are responsible for carrying information about traits between generations. And this is also referred to as particulate inheritance. Or this phenomenon is referred to as particulate inheritance. And so again, scientists knew this existed, but they didn't know what molecule was responsible. So DNA is found in almost all cells, so that was a good candidate to be the heritable molecule. But the DNA structure was pretty uniform, which we talked about earlier. And scientists, some researchers actually saw this as something that would not be a character of a heritable molecule. And also we know that DNA only has these 4 different bases or 4 different nitrogenous bases. And scientists said, how could we make up all of our traits just out of these 4 different bases? It seems like there's not enough variety in the monomers. Proteins, on the other hand, came in a variety of sizes and shapes, and there are 20 different monomers. They're also found in all cells. And so it seemed like proteins were a pretty good candidate for a heritable molecule as well. So how did scientists actually determine whether DNA or proteins were carrying genetic information? Well, they did a bunch of experiments pitting proteins and DNA against each other. But first, they needed to prove that there's actually some molecule that was being transferred that was carrying information. And so this is the first experiment we'll talk about. These are the Griffith experiments. And this is in your textbook, and again, the diagrams are a little bit different. This is from another textbook that I use, but it has the same information. And so the basis here of this experiment is that these are scientists using two different strains of bacteria. One, which was pathogenic and eventually fatal to mice, and another strain of bacteria that was not fatal to mice, not pathogenic. And so these two strains of bacteria were really similar, but they knew that when they grew them up in a petri dish, they looked a little bit different. The surface of the bacterial colonies looked different. And they knew that these two different types of strains had different effects on mice. So the one that they designated that they knew was pathogenic, as we said, if you inject it into mice, then it ends up with the mouse dying. And this was referred to as the S strain. The other strain, referred to as the R strain, when you injected that into mice, then the mice were fine. But scientists also found that you could kill the S strain. So if you killed the S strain and then injected that solution into mice, then the mice were fine. And they would kill the strain by heating it up. So again, it's just similar to sterilizing something with heat. You're still going to have the dead cells there, but if you inject dead cells into an organism, usually you don't have that effect. However, if you took a live R strain, which is non-pathogenic, that's the little one there, and you mixed it up with the dead cells from the pathogenic strain, and you put the mixture and injected it into a mouse, then it had a pathogenic effect. Now, this is counterintuitive because the cells that are active and alive should not be pathogenic. And so scientists said, okay, what's going on here? So what happens is that the information on being pathogenic and infecting a mouse is transferred from the dead cells to the live cells. So we know from this result that there is some type of heritable information that is passed from the S to the R strain, and there is some molecule that's carrying that information. What we now know, and what's really neat for me as a microbiologist, is that some bacteria, when they're stressed, will pick up DNA from the environment. Not all bacteria, but some bacteria. And so this is one of the reasons why we had a lot of outbreaks of MRSA, maybe 10 years ago, a little bit less, because some bacteria can just pick up DNA from the environment, from the dead cells around them, particularly when they're stressed. And so they could pick up this DNA, and then they can use the DNA to make proteins if it's from other bacteria. So in this case, it allowed the R strain to make pathogenic proteins. In our cases, then some of the MRSA would pick up proteins or genes for proteins that are resistant to antibiotics, that antibiotics can't affect. Okay, so we know that there's some kind of information being transferred, probably through a molecule. So let's fast forward 15 years in the future. Different scientists, different labs were using similar kind of setup. So again, we're looking at these bacteria, two different strains that are similar, having two different appearances of the bacterial colonies there. The rough colony is on the left. You see it's kind of wrinkled, and then the C colony or the S strain is on the right. And now scientists were able, they had the technology to separate different types of molecules. They still didn't know the structure of the molecules in great detail, but they knew that there were different densities to these molecules, different weights, or different masses, and so they could separate them out by killing the cells, breaking open the cells, and then centrifuging them, or separating them by their density. So what they did was to have a bunch of different experiments where they took the different molecules from the S strain, which is pathogenic, and they added those different molecules to the R strain, which was non-pathogenic. And they did each of these in a different experiment. So in one experiment, the non-pathogenic strain got RNA from the pathogenic strain. And in another experiment, the non-pathogenic strain got protein from the pathogenic strain. And then they mixed these up, injected them into mice. And what they found was the only experiment that resulted in a pathogenic form of the bacteria was mixing the DNA from the pathogenic strain with the non-pathogenic strain. So this supports the hypothesis that DNA is the variable molecule, right? Because in these experiments, we mix the protein with the live strain, the mouse lives, no pathogenic effect. But other labs did not believe them because they were so committed to this idea that proteins were the variable molecule. They said, no, you contaminated this experiment. You didn't get all the proteins out, and that's why the mouse died. Okay, so then another experiment was done, a follow-up, and they actually did the same experiment, and they added a certain type of enzyme that destroys DNA, and the mouse survived. So that was even more concrete evidence that DNA is what is carrying this information, and that is what is being transferred to the non-pathogenic strain. But of course, some people still didn't believe that. So we're fast forwarding again to the 8 years in the future. Where she indicates in this experiment. This is really cool. Now we get to talk about viruses and bacteria. So viruses are technically not living organisms. They're not made up of cells. And this is just one form of virus. This is a bacteriophage. It infects bacteria. It doesn't infect human or eukaryotic cells. But in general, viruses have very few parts. They have protein usually and DNA. Some of them also have a membrane, but this type of virus does not. And so scientists knew that, they didn't know the structure that we're seeing here, but they knew that there was protein and they knew that there was DNA in a virus, and that was it. And they also knew that these viruses could infect bacterial cells. And the whole thing about viruses is that they infect cells to make more viruses. So I'm going to go out on a limb here and be a real nerd and say, who is a Star Trek fan here? If you're a Star Trek fan, you know what the Borg are? This is really similar to the idea of the Borg. They are this society that kidnaps other organisms and makes them into more Borg. So this is basically what the virus does. It infects the bacterium with some kind of information, and then it forces the bacteria to make more viruses. Yes?
So, basic part, pieces to put itself together, and it makes it.
So what's going on here is that we've injected the information molecule. And then the cell is using that information molecule, which we know to be DNA, to make new proteins and new DNA instead of the cell making its own protein and DNA. It's making viral proteins and DNA. And then With this type of virus, it lyses or breaks open the cell, and then the viruses leave the cell.
It's the lytics.
This is the lytic cell. So some viruses don't break open the cell, and those are viruses that tend to perpetually come back throughout their lifetime. Okay. So why is this important? Well, because we know that there are only two types of molecules in a virus, and we know that one of them, the information-carrying one, gets injected into the bacterium, we can label these two types of molecules differently. And so the idea is to figure out where the two molecules are. Are they inside of the cell? Are they outside of the cell? Kind of like using trackers if you're following migrating animals. Only in this case, you're not using a tag or a radio collar on an animal, you're using the radioactive atoms in one of the molecules. So for this experiment, in order to label only one molecule, either DNA or protein, you need to label elements that are only found in one of the molecules. So in the case of DNA, the scientists label phosphorus because phosphorus is only found in DNA. And as we said, there's lots of phosphorus in DNA because of those phosphates. In the case of proteins, we don't have very many unique elements that are different than DNA, but proteins, cysteine and methionine in particular, these two amino acids, contain sulfur. And so the scientists also knew that the viruses they're looking at had proteins that did contain these amino acids. And so they were able to label these two amino acids with sulfur, and they're able to label DNA with radioactive phosphorus. How does this occur? Well, I say it as if it's a very specific kind of process, and scientists are going around and removing atoms and replacing them with radioactive atoms, but that's not what's happening. All they have to do is add radioactive sulfur to the solution where they're growing up bacteria with viruses, and then those radioactive sulfur atoms are going to be used in place of normal sulfur atoms. And similar with the radioactive phosphorus for DNA. So the scientists would make up these viral stocks that either had radioactive sulfur or they had radioactive phosphorus. And remember, keep that in mind because a lot of people get that confused. They do two different experiments. They had viruses with radioactive sulfur and viruses with radioactive phosphorus, no viruses that had both. So then when they were ready to test whether DNA or proteins were being injected into the bacterial cells, they first took these bacteria that, these viruses that were labeled with radioactive sulfur. So are we labeling DNA or sulfur, or DNA or amino acids here? DNA, or with sulfur, that's the amino acids, methionine and cysteine. So sulfur, we're labeling amino acids. So these phages, these viruses, only had the amino acids with proteins labeled. The DNA was not labeled. This whole process where the viruses go through this whole process. And then they actually put these guys into a blender to shake off whatever pieces of the virus were on the surface of the bacterial cell. And then they separated out the components to see what was associated with the bacterial cell itself. And so what they found, and I think it's a little bit difficult to see in this image number 3, but the red part on top where the virus particles end up. And that's where they saw radioactivity. Which means that since the amino acids were labeled, the amino acids were radioactive, and they were only in the top layer here, which is where those viruses particles were, that were on the outside of the cell. The DNA, which got injected inside of the bacterial cell, was in the bottom, wasn't radioactive. And then second experiment, we do the same thing, but with the viruses that have radioactive DNA, which has radioactive phosphorus. And in this case, again, they knock the viral particles off the surface of the bacteria, and then they separate this by density. And there's no radioactivity in the top here, where the virus particles that were on the outside end up. But in the bottom here, what's called the cell pellet with the bacterial cells, they see radioactivity. So we know that that radioactivity is caused by the phosphorus, which is in DNA. So we know that the viral DNA got injected into the bacterial cell. Which means that that is the information that's being transferred and used to make new proteins. So this is the heritable information.
Just the viruses and the protein.
So what went into the blender was the bacteria that had been infected by the viruses. So they contain both protein and DNA. And if we go back to this here, so you see that there are these different steps here. So it was important in this experiment that the scientists didn't let it get to this step where the viruses were going to be released. Because if you allowed it to get to this step, when it had the radioactive phosphorus and the radioactive DNA, then the DNA kind of new viral particles. The cell would be burst, and so those would also not be in the cell plate.
So,
So it's in separate.
yes, so with design, did it just prove that DNA has the information and the protein makes whatever the new viruses?
So they wanted to figure out what goes inside the cell in this first step. Is it DNA that goes inside of the bacterial cell, or is it protein that goes inside the bacterial cell? So they labeled either DNA or protein, and then they let it get to this step, and then they saw what was inside and what was outside. So the thing that's missing here is that it's not showing that that viral particle is still on the outside. So the protein that was originally made protein when that started out will still be on the outside here, the DNA is inside. And the new proteins that are made are not incorporating radioactive ions.
So they're kind of like,
So right, yeah, they're not ready to infect the cells yet. And as kind of a follow-up experiment to prove that indeed there was viral DNA, there was viral information that was put into the bacterial cells, then after they measured radioactivity, they allowed the experiment to run a little bit longer, and they found that the bacterial cells were lysed, and then you could see that, or you could take that solution and infect new bacterials with that. So it did contain functional viral particles. Any questions about this? So they discovered that the DNA that was injected, so they basically found out in this stuff that DNA was injected. And so they could say, oh, but was it actually going to be used? And so then they said, okay, we want to experiment on lumberers. It is used to make new viral code. And so that's a pretty good point. So then what they could have done is experiment at the top, and it's they could have separated the red solution that was radioactive and had proteins and just taken it out as only DNAs themselves, and that DNA was still used to make new viral proteins. It didn't need those protein shells. Move them and it's okay, as long as the DNA is already moved out. Any other questions about this? And this again is definitely something that I would advise that you diagram out to make sure that you really truly understand it, because I think that it's easy in some ways to feel like you've got the takeaway, because of course DNA is what is carrying information, but this is kind of like deriving a mathematical formula or property. You really have to understand every step to understand why these results prove that DNA is carrying information. So we'll talk a little bit now about DNA on a larger scale. I think this was a nice transition because we talk about an entire genome of virus being injected into a bacteria. But viral genomes are really, really small compared to bacteria or humans. So on this image, I just love this chart for so many reasons. Here we see the average genome size of a bunch of different organisms. So at the bottom here, we see that relatively archaea and bacteria, the prokaryotes, have fairly small genomes. And then as you start to look at all the eukaryotes, first of all, there's a huge diversity of genome sizes. And also, what do we expect to see? Where do we expect humans to be on this spectrum? We expect to have the biggest genomes, right? We don't have the biggest genomes by a long shot. So we're here, we're kind of in the middle. So this kind of indicates a couple of things, that more DNA doesn't necessarily make for a more complex organism. And it also speaks to the fact that throughout evolution, certain organisms, in particular animals, tend to lose DNA that they don't need. I guess not even animals, really just mammals, birds, and reptiles. So there's also a lot of this DNA. Fishes and amphibians carry around a lot of excess DNA. And then plants. Plants are very interesting because they could actually have duplications of their entire genome. And that's actually one of the things that we as humans cultivate in plants. Because DNA, we have so many nucleotides. If a plant genome is duplicated, and they have twice as many nucleotides, they actually end up having larger cells, and that's really good for farming food and for agriculture. And so we haven't necessarily done this knowingly throughout history, but if you look at the genomes of things like, I think it's happened in bananas, for example. They have extra copies of the genome. So that's one of the reasons why bananas don't have, are not functionally reproductive. So bananas don't have seeds. You can't plant a banana in the backyard. part, I think, because the genome had been duplicated. So anyway, how do we fit all this DNA into the cell? On the left here, we see that all this DNA is surrounding this E. coli cell, and this cell has been punctured. All this DNA is moving inside of the cell. So how do we fit all that DNA inside of the cell and also keep it organized somehow so that the right genes are being expressed and that so that we could replicate the DNA? So this is a complicated system that needs to do a bunch of different things. We refer to this as DNA packaging. We need to consolidate the DNA. But in addition, it's not like when you go on vacation and you just sit on the suitcase in order to close it. You can't just throw DNA into a really dense ball. You need to have some kind of system where some of the DNA is accessible, so that, and also a system where that gets somewhat organized, so that different proteins can identify different regions of the DNA. And of course, we don't want to get the DNA tangled because we don't want to break the DNA. And so this is a really nice image showing up at the top. This is more densely packaged DNA in this darker region. And then the lighter regions are areas of the DNA, of the genome, that are less densely packaged because they're used very frequently in the cell. And they're actively being used either to copy DNA or to express genes. And so this is what we would normally see in most of the cells in your body. They're using some DNA, not all of the DNA in the genome. And so some regions are more densely packaged than others. But when we think of our genomes, we often think of something like this. These X-type structures. And these are actually chromosomes. And that level of density is only seen in the cell before mitotic division. So if your cells are dividing, then you would see the DNA in these structures. Otherwise, it's not going to be that densely packaged. But most of us, if we've seen DNA under a microscope, we've seen it in this form, in the mitotic chromosome. And we'll see it next week, I think, when we talk about mitosis. Okay, so how do we organize the DNA? So there are lots of levels of this, and I can't remember if your textbook talks about every single level, but I'm going to focus mainly on this level, the DNA double helix wrapped around these proteins called histones. And just focusing on the DNA wrapped around histones, this kind of structure is called the beads on a string model. It looks kind of like beads on a string. And each one of these units is called a nucleosome. So a nucleosome is that portion of the DNA wrapped around these proteins. Kind of looks like a little hedgehog or something. And so this is more of a cartoonish depiction. Again, we have a region of double-stranded DNA, and then that is wrapped around this nucleosome, or wrapped around these proteins, so that we get this kind of beads on a string, these little disc-looking things, disc-looking structures. And then these can be organized to higher and higher levels and stack up so that they form more and more dense structures. So I think we're going to see that. And histones are the type of protein that we find inside of these nucleosomes. So again, with the nucleosomes, the nucleosome is these proteins and the DNA wrapped around the protein. And if you want to be completely accurate, I guess we would draw the double strands of DNA. So there's two loops of your double-stranded DNA wrapped around each of these sets of histones.
And that's the same nucleosome is with which we call this.
So the nucleosome refers to the histones with the DNA. Now another term is octamer. An octamer refers to just proteins or just the histones. I try and draw it in a 3D way. I apologize if my art skills are subpar.
Can you just say one more time what the strands are again? And what the exactly what the nucleus, so the nucleosome is like actual ground.
Yes, so the nucleosome refers to DNA plus proteins, whereas octamer refers to proteins only. Yeah.
So this one is
Yes. And so again, we get the group is octamer of 8 proteins. And as we said, these are histone proteins, and they're often labeled as H1, H2, H's and different numbers. But we have duplicates in this octamer.
so it's not 4 types of proteins, it's 8.
It is only 4 types of proteins, but 2 of each. So in this octamer, we have H2E in front here, and then we have another H2E in there. And then similarly, we have an H2A, and we have another H2A. So forth. So that we have 4. So we have 4 different types of proteins. And as we said previously, these proteins, these histones, have a particular type of amino acid. What do we say the characters of these amino acids have? So what type of amino acids do we see in these histone proteins so that they're really good at interacting with DNA? Positively charged amino acids. Not only positively charged amino acids, but there are lots of positively charged amino acids that interact with attract the negative charges of the phosphate groups on DNA. And so this is important to remember that we have charge attraction here. There's no covalent bonding between the DNA and the histone proteins. So the interactions are relatively, they're weaker than covalent bonds, but they're still stable. Your DNA is not going to just unwind spontaneously from the histones. But this does mean also that we can remove the DNA from the histones without breaking any bonds. And so we'll come back to this in a minute. I'm missing a slide here. So I think in your textbook it talks about certain proteins that will interact with these histone octamers, or really with the entire nucleosome, and it can slide the DNA so that different regions of the DNA are going to be interacting with the octamer. And so that frees up the DNA, because we go back to this image, we see that there are regions here that are not associated with an octamer. And so these regions of DNA between the nucleosomes are more susceptible to damage, but they're also able to be read and interacted with by enzymes. So DNA that's wrapped around an octamer that's part of a nucleosome is not going to be read, it's not going to be replicated, it's not going to be expressed. So if you want to express DNA, you have to remove it from that octamer, but you're not going to take it and unwind it and unwrap it off the octamer. You're just going to slide the octamer to a different region of the DNA. So only the DNA is being read. And again, that linker DNA can be read, it can be bound by other proteins, and it actually is susceptible to damage by certain enzymes as well. Is that clear? And we'll talk more about this when we talk about gene expression. I did want to talk a little bit more about the histones and how the histones play a role in identifying DNA. Now, I feel like there's going to be not many people that raise their hand. Who remembers card catalogs? Card catalogs and libraries? Yeah, okay. How many people take these and how many people feel so ill? But these were these things where in the library you had a label on the front that had the part of the alphabet that this was referring to, and then it had a list of hundreds of books that either had a title under those letters or had a subject relating to that. Yes, so that's how you used to look up things in the decimal system. Yes, exactly. And so before you pull that drawer out of hundreds of titles and topics, there was a little placard, like I said, on the front that said, okay, this is, numbers in the Dewey Decimal System 1 through whatever. And so that would tell you what was in there before you opened it. And so we need a similar system for DNA because enzymes can't go through and move aside each octamer in order to look at DNA and see if this is the gene we need to express. That would take forever. It's not useful at all. Instead, what happens is the histones are actually modified chemically so that they have some kind of indication as to what DNA they're bound to. So these are called chemical groups or chemical modifications that are on a certain part of the histone called the histone tail. So for simplicity here, I just drew these spheres, but in reality, each of these has a tail. And the tails project out so that other proteins can interact with them. So I think this is a really nice image. The gray that's wrapped around the octamer is the DNA. And we can see that each of the histone proteins has a tail that projects out from this nucleosome, so they can be read. And again, keep in mind, what are the histones made up of? Amino acids. And so there are different amino acids in the different histones. So each of the tails of the 4 different types are different. And the amino acids can have different chemical groups added onto them. So remember how we said a couple of days ago that things like methyl groups or phosphate groups can send a signal? This is one example of that. So other enzymes can come in and they can add a methyl group to some specific amino acid on one of the specific histone proteins. And that, in addition to all these other chemical groups added onto the same or other amino acids, you see here there are lots of chemical groups added on. This pattern is the indicator for what kind of DNA is wrapped around this particular nucleosome. So it's not totally unique to the DNA. We might have multiple nucleosomes that have really similar tail modification patterns, but this would send signals such as, for example, this DNA should never be expressed again. And we have DNA that is expressed at one point in our lifetime and is never expressed again. So yesterday we were talking about the globin proteins. Now we have a certain type of globin when we're babies. There's another type of globin that's made from a different protein, but we never want that gene expressed again when we're adults because that's only useful when we're very young. And so in that example, that globin protein would probably be part of a nucleosome that had labels on the histone tails that said never express this DNA again. And this is all these chemical groups and these patterns of chemical tags are read by other proteins. Yep. So the tails are just strings of amino acids. Yeah. So if we think of it again, proteins made of amino acids, we make this long string of amino acids. Part of it folds up into this kind of globular part of the protein, and then the rest of it is the tail, still amino acids.
And that's what the chemical groups are attached to.
Yes. Yes. And the chemical groups are attached covalently to the tail. And as we can see, for example, right here, some amino acids can have multiple chemical tags on a single amino acid residue. And that's kind of like doing multiple locks on the door. So this would send kind of a double signal. Don't express this DNA. So, in addition to sending signals, or sending signals about whether or not this DNA should be expressed, the chemical groups also send signals for how dense this DNA should be packaged in terms of the higher structure that we're not going to talk about too much. But we could see, you see here that the histones can be stacked in rings, and those can be stacked on top of one another in various levels of density, all the way up to the most dense level, or most dense form, which is the mitotic chromosomes. And again, the histone tails modifications help to indicate how dense each region of DNA should be. So in the opening slide here, the nucleosomes, or the octamers, the tails of those octamers would have different chemical tags here than here, because the signal for these guys would be to be more densely packed, and these guys would be less densely. Any other questions on this? So we're gonna try to remember today or tomorrow to send out the link. I have a Google poll site where you could submit an anonymous question. So I think this will be really good for us for over the weekends. So if you guys are going over things, come across something, and say, oh my gosh, I'm going to forget by the time Monday rolls around, just submit it on that question site, and then we'll try and go over those at the beginning of class on Monday, or at some point on Monday. But unless anybody else has questions, we're going to move into the lab, do the problem session as well as the lab. I just did want to make one more note, or two notes. We will have a quiz on Monday on amino acids and proteins. And keep in mind the monomers versus polymers. And then I will also post homework tomorrow that will be due on Monday. It'll probably be online. I'll send out an email when I can post. And I'll give you a couple of guided topics, like 3 or 4, like I did from the previous quiz. Was that helpful?
Yes.
Okay. Yeah, definitely do that.
Then you know what exactly what to study.
Not exactly. I can definitely do that. All right. Any other questions before we move into the other room? We'll have a 5-minute break so that you guys can get your stuff together.
You said the quiz is about amino acids and proteins.
Yes.
And then the difference between.
Amino acids and proteins and DNA. Did I say
And
that?
DNA.
Oh, I said amino acids and proteins. I meant amino acids, proteins, nucleotides, and DNA.
That's why